This lecture is about the biology of propagation, the background information, more the science behind plant propagation and inducing plants to grow the way you want them to. So I'll be covering a number of the topics included in the biology of propagation, and I will include the information that's the most important here and not go into super high detail in all of these subjects, but cover the basics for you to understand. Let's get started. So some terms to know about, thinking about more the biological aspect of propagation, not so much the business of it, but biology, are the genetics. The term genotype means the genetics of that organism. So the genotype is their genetic makeup, the DNA and all the genes that are inside every cell of their body and what those code for. The phenotype, in comparison, is about the expression of those genes. So phenotype is kind of like the overall appearance of that organism. The size, the vigor, the color, the shape, could be the biochemistry of that organism. Those are all the ways the genes are expressing themselves physically in an organism's body. That's the phenotype, is the way those genes are expressed. And the genotype is just the genes themselves. So that's the difference between genotype and phenotype. You might say, well, you have the genotype for blue eyes. And that would be, because someone's looking at my blue eyes, but they would also know that I have that phenotype. So I have the genes for blue eyes, and I also have the blue eyes that go along with those, the expression of those genes. And any discussion of the biology of propagation, since propagation is about creating new individuals of a species. So if you want to propagate roses, you're making more rose plants from one rose plant by cuttings or seeds or something like that. All of propagation involves cell division. So new cells have to be made, new tissues have to be made, or new seeds have to be made to propagate a plant. So I want to talk to you about the two types of cell division. Mitosis is what you're seeing here, and that is the cell division process where one cell, you can look at the image if you want in the upper left, one cell is undergoing the duplication process. That cell is dividing and turning into two cells. So one cell divides and becomes two cells. That's how all living organisms grow. They grow new cells by an original cell duplicating and turning into two. You can call that the miracle of life, or you can call it mitosis. And in mitosis, the cell that divides to become, that was originally one and becomes two, is identical genetically and phenotypically, but pay attention, mostly the genetically is identical to the original cell. So mitosis is just making copies of new cells for that plant, for example. It could be an animal, but in our case, for a plant, it's just grow. You have your rose bush gets taller and taller. It's adding new cells to its tissue by mitosis. That's one cell division process, which is mostly how all organisms grow. Mitosis is the form of growth and reproduction that's used in asexual reproduction. So if you are layering a plant or taking cuttings or little bulblets or pups grow off of an agave, that's asexual reproduction. There's been no formation of eggs or sperm, and there's been no sexual fertilization. It's just been growth of new little clones of itself, either human-induced or natural, through mitosis. It's called vegetative propagation, and results in the same number of chromosomes in the original cell as in the resulting cell. For example, a plant in the genus Prunus, there's many, many species in Prunus, they have multiples of 8 chromosomes. The peach and apricot have vegetative cells with 16, cherries have 24, plums have 32, and through mitosis, the cells that are created in mitosis in those Prunus species would have the same number. So a peach would have 16 chromosomes normally, and as it goes through mitosis, the resulting new cells would have 16 chromosomes. And that's different than meiosis, where the chromosome number changes. Where is mitosis happening? It's in the meristems. It's kind of like stem cells in animals, but the meristems are the places on the plant's on the body of the plant where cells are dividing and making new cells. They're adding cells to the body of the plant. So mostly at the tips of the branches, like you're seeing here on the two right pictures, or the root tip. Those are places, roots are growing down deeper and pushing into the soil, so they're elongating, they're adding new cells so they can grow. And the same with branch tips. They're growing up, reaching to the light. They have to add new cells to get taller and bigger. That's where mitosis is happening in the meristems. Okay, so mitosis and growth is happening in the meristems on the tips of the roots and the tips of the branches for the most part, but where specifically in those places is it happening? It's happening in the vascular cambium. The upper left picture is showing you a diagrammatic illustration of a cross-section of a stem. Could be a branch of a plum tree, lots of different things. And inside what we would call the bark, they're showing you there the corky layer, cork cambium. Those are more detailed than you need, excuse me, than you need to know. But inside the bark is a thin layer called the cambium. And that cambium is the place of cell division. So if you looked at the whole branch, it would be like a cylinder that's just really one cell thick underneath the bark, going all the way up the branch or the trunk of that plant. That is a meristem. That is where those cells are dividing to make new cells. So earlier I mentioned there are two types of cell division. One is mitosis, where cells are making exact duplicate copies of themselves. That's mitosis. The other cell division process is called meiosis. And in that process, the cells are dividing, so new cells are being made, but the end product, the cells that result from this process, only have half the number of chromosomes of the original cell. So in meiosis, you start with one original cell which divides, it actually divides again, and you get 4 resulting cells that each have half the number of chromosomes as the original cell. Compared to mitosis, that cell divides and makes 2 resulting cells that have the same number of chromosomes as the original. So in meiosis, things change a bit. Meiosis is the cell division process that only is happening in the reproductive organs of an organism, where the cells are getting prepared for sexual reproduction. So in other words, let's say in a cat, meiosis is only happening in the ovaries of the female cat or the testes of the male cat, where eggs and sperm are being made. Eggs and sperm are the result of meiosis. So you have a cell that's kind of like a pre-egg or pre-sperm cell. It divides once and then twice again to make 4 sperm that each have half the number of chromosomes. So if you think about the kind of longer goal here, eggs and sperm are being made by meiosis. Even in plants, eggs and sperm are made. So they have to have the number of chromosomes, because then when a sperm and an egg get together later, hopefully it's been crossbred, then it goes back to being the normal number of chromosomes. So the eggs and sperm have to have half the amount, because an egg and a sperm unite. You've got half of the number of chromosomes in the egg and half the number in a sperm, and those get together. Now you have the whole number, back to the normal number of chromosomes in that organism. So in a cat's body, the hair on that cat, the ears, the eye cells, the bone cells, the muscle tissue, the organs, they're all being grown and replaced through mitosis, where exact copies are being made, except where eggs and sperm are being made. That's meiosis. Same in a plant. So the plant's body, it's growing by mitosis on the stems, the leaves, the roots, the flowers or the cortex, except inside the flower where pollen is being made, which has sperm, and the ovules are being made, which are eggs. That's where meiosis is happening, inside that flower, the reproductive parts. But the rest, even the petals of the flower, it's all mitosis. In meiosis, that is something that's happening when you're propagating things by seed and you get seedlings. They have gotten a sperm and an egg, have united from different sources, maybe the same flower, maybe a different plant, but each egg and sperm are genetically unique. So you get unique genetic individuals and seeds with different genetics when it undergoes sexual reproduction through meiosis. Flowers have the sperm and the pollen, as I said, and ovules are produced that have the eggs in them in plants, and each is what we call haploid, meaning having half the chromosome number as the originating cell. And again, that's to prepare. You have half the chromosomes in the sperm and half in the egg. Fertilization happens when those two join, and you get what's called a zygote, which turns into the seed, which has the full complement of chromosomes. And because the seeds have different genetics when it undergoes sexual reproduction involving the cell division process of meiosis, the seedlings will have different morphological appearances. They'll look, their shape, their color, their size will be different. Physiological characteristics could be different. And the way they interact with the environment could be different. And this is the whole let's see, what would we call it? Not really a motivation, but this is the whole way that evolution works, is that we have a population of one certain species, and each is genetically different and physiologically different and morphologically different, interacts with the environment in a little bit different way. Thus, some are more successful, some are fitter, they do better in a certain environment, they handle the weather better or the amount of water, so their shape is different, their physiology, so some do better than others. So that's what survival of the fittest is Darwin's idea that some individuals in a population genetically will be more successful at survival and reproduction, and those ones are the ones most likely to go on and give their genes to the next generation. So over a long period of time, you get the individuals in a population are more and more honed to survive there better. And over even longer, much longer periods of time, that species evolves to be more adapted and focused on survival in that area. So your genetic pool gets more focused on more success and reproduction. So if you had everything was a clone all the time, and let's say the environment changed, imagine a population of agaves in the low foothills of Baja California. They're wild. And as the temperature, maybe the weather and the climate has been changing over the last 100, 200,000 years, it's getting warmer and warmer. If all of those agaves were exact clones of each other and they were starting to have negative effects from increasing climatic temperatures, they would all respond the same way. They would all be damaged. They would all have negative effects, and at some point they might all die. But if you had a variety of genetics and physiologies and morphologies within that one species, just like we do in a group of people, we're all different, then some will survive that and some won't. And those are kind of like evolution's hedging its bet. Some will survive and make it and adapt to the new changed climate or environment and go on to spread their genes, and that kind of replaces the less adapted one. So that's the way it works. So overall, how do plants grow? By cell division, mitosis, cell enlargement. The cell doesn't have to divide to get bigger, it can actually elongate and kind of stretch out. And then cell differentiation. Some cells will become a specialized cell, and they might get a little bit bigger from like that cambium cell that divides. It could become a xylem cell or a phloem cell. If it becomes a xylem cell, it's going to end up being larger than the phloem, and there's other examples too. Some other terms you can come across talking about the more biological aspects of propagation are totipotency, competency, and determinism. Totipotency means that each living cell has the potential to reproduce the entire organism. This is an important concept when you're working with tissue culture, when you take just a little bit of a part of a plant, like a part of a stem tissue or a leaf tissue, and through hormones and kind of artificially, we can induce it to produce a whole new plant. That is because of totipotency. It's because each cell in a plant, and almost all organisms, has all the genetic material to code for the whole species. Even though you might take a little bit of tissue off of a flower petal, every cell in that flower petal has the same DNA that any other cell in that plant does that codes for everything, not just the flower. It codes for the root strength, the root length, the xylem cell growth, the epidermis, the leaves, how tall it's going to be, what color flowers, everything in that one cell. That means each cell is totally potent, meaning it has all the info. And with humans, what we'll do is we'll induce certain genes to be turned on and off using hormones if we're doing tissue culture to get that one cell to grow all parts of the plant. Competency is the potential of a given cell or tissue to develop into a particular way. So some are more able to, even though it has all the DNA, it's totipotent, it doesn't mean that it will or can develop into a certain tissue. Some genes will remain turned off in a stem tissue. If you took a little piece of stem tissue, it might be very difficult to get it to, or impossible to get it to produce roots. So competency is talking about its actual real potential to develop those. And it has more to do with how easy it is or how hard it is to get a certain piece of tissue to develop new types of tissue on it. Determinism is how committed the cell is to produce specific organs. It usually has something to do with a temporal factor, meaning if it's starting to leave the meristem tissue stage, where a meristem cell, just like a stem cell in animals, a meristem cell still hasn't differentiated, hasn't gotten a certain role yet, and it can become anything. But once it starts to change into a stem cell or a leaf cell or a root cell or a flower cell, flower petal cell, its determinism is increasing. It's becoming more specialized already. In genetic engineering, DNA from two organisms are joined. So usually a third organism is the vector, meaning these two bacteria listed here, these agrobacterium, are often the thing that DNA is taken from one organism, placed into the genetic material of this bacteria, then which is bombarded or sprayed onto cells of a plant, which take in some of that some of the DNA from that first organism. So you've got two different pieces of DNA, usually both from plants. You've got your host plant and you've got some DNA from another plant. You want to get that piece of DNA from the other plant inserted into the host plant DNA. And this bacteria is the vector. It carries it really from the donor DNA plant to the host DNA plant. Sometimes the DNA can be isolated from that donor plant and made into little gold pellets, which are shot into the DNA of the new recipient plant. Sometimes the agrobacterium is the one that kind of carries it over and gives it to the recipient plant. It's all something that produces what are called transgenic plants that have DNA from two different organisms, or recombinant DNA technology. It's what GMOs are. Genetically modified individuals are done through this process, which involves tissue culture. Two methods that I mentioned, the gold pellets, they can be shot onto the DNA of that recipient organism with something called a gene gun. And it ends up, this is an example of one, with transformed embryos that have received DNA from a different organism to give a transgenic rice plant. Plants produce hormones which control the growth of that plant, so I'd like to now mention a number of different common hormones in plants that are controlling their growth. We've talked about this one a few times, but auxins are one that is a type of hormone that's made in developing leaves of plants, and it moves from the canopy, the leaf canopy, down to the bottom in general. It inhibits lateral or side growth, so it keeps apical dominance, meaning auxins are produced and it makes the plant stems grow tall and upward. It can induce roots and adventitious root growth on the cuttings. IBA is one of them that we've used for our cuttings to induce rooting on. Auxin stimulates adventitious rooting, and as was said a little moment ago, it suppresses lateral growth. So if you cut off the tip of a stem, the lateral sides shoots start to really take off because the apical dominance caused by auxin has been removed. This is a picture showing you of a bean plant with the stem tip intact where auxin's being made, and these side shoots, they call them axillary buds. They're these lateral shoots found right kind of in the armpit there between the branches and the main stem. Those are not growing. They've been suppressed because the auxin is being produced in the tip of that stem. In here, the shoot tip has been removed, and now those lateral buds are not being suppressed. Auxin is not being produced as much there. It's not suppressing lateral growth, and the lateral or axillary buds take off. Cytokinins, they are another type of hormone. They are essential for cell division. Kinetin was first isolated from fish sperm and shown to promote cell division, as zeatin from corn was also taken out and put on other plant cells to show that they induce cell division. This is important for plant growth in many ways. The interaction of auxin and cytokinins is also kind of interesting. The ratios, if you have a high amount of auxin and low cytokinin ratios, it induces root growth. The opposite induces shoot growth. If they're both high, it produces more of a callus growth that we've seen in our cactus and succulent propagations, where it forms kind of scabby material and plugs up the wounds. The mode of action of these isn't known, so there's still more to find out. So from these little cell blobs, these little kind of like tumors of cells on a petri dish that are grown from this plant, they have high auxin, low cytokinin. Hormones are inside this petri dish, so they were artificially put there, and it induced root growth. That's one of the ways they found out the ratios and how they induce different types of growth in those hormones. What kind of ratio of auxins to cytokinins would give these leaves a shoot growth of this little cluster of cells in a lab situation? It would be the high cytokinin, low auxin ratio. Gibberellins are another main category of hormones found in plants. First discovered on abnormally tall rice plants. People were wondering in Japan, why are these so tall? What is it in them that's causing that? It's caused by, well, in this situation, it was caused by a fungus that made the plant overproduce this gibberellin hormone and made it grow tall. It's transported in the xylem and the phloem. It's found in seeds and controls seed dormancy and controls germination as well. It just promotes shoot elongation, as you can imagine looking at these abnormally tall rice plants. So cell division and elongation of those cells. That's what gibberellins are for. When I first learned about these two, when I went to school at UC Davis, I was taught that these are something that's either given to or is inherent inside plants that are bolting. If you don't know what that is, it's like when you grow things like spinach or lettuce, cabbage, things that are kind of supposed to be low growing and leafy, and that's what we want to eat the leaves. But at some point it gets warm enough and they get old enough and they produce a whole bunch of gibberellins and they send up this big, tall, flowering stalk, and then you can't eat the leaves anymore. They're too bitter. So it's called bolting, when it shoots up the stalk to flower, and that's when gibberellins are being produced. And you can induce bolting by adding gibberellic acid or gibberellin hormones to your plants if you wanted to. This has just been done in a lab situation. People wouldn't do that in their garden, but just so you know. There's also abscisic acid. It's naturally occurring. It's a growth inhibitor. It controls the dormancy of buds and seeds and leaf abscission. That's the main thing I remember from being an undergrad and learning about this was that it's being produced in the fall to make leaves, like deciduous tree leaves, have a kind of a clean cut between them. Why do those leaves fall off? Because abscisic acid makes a little cleave, like these little cells seal themselves off from the mother plant until there's like a little break between the tissue between the branch and the leaf, and that little break opens up. Abscisic acid is driving this, and then the leaf is at some point not even connected to the plant and falls off. Ethylene is a gas. It's produced in fruits and flowers, and it causes the flower death or senescence. It can also induce flowering. It can induce adventitious fruiting. The main thing it's known for is it causes ripening and fruit maturation. That's why if you have fruit and it's not ripening fast enough like avocados, you put them in a bag, because it's not because they like dark and they like to be in a bag. It's because ethylene gas is produced by the avocados and it will concentrate. It won't just float off like it would on your counter. It will concentrate in that bag and hasten the ripening of that. And that's something that has to be controlled in fruit that's being transported long distances. If you have a huge truckload of tomatoes, they're all producing ethylene and they're all making each other ripen, sometimes way too fast. If you want bananas to not ripen so fast, split them apart from your bunch of bananas and keep each banana, a couple feet away from the other bananas. If you have them all spread out on your counter, it would look really weird, but they would ripen much slower. It's the ethylene gas that they're producing that they ripen themselves is what these fruits are doing, getting them ready for being eaten. So that's what sugary fruits mostly are, at least, historically, before humans manipulated them a lot, they were made for animals to come eat them and carry the seeds inside of them away to be planted somewhere else. Here's some more information. We're moving off of the topic of plant hormones now, and just some other bits and pieces of information about the biology of propagation. This is a review, hopefully, different types of life cycles of plants. We have annuals that complete their whole life cycle in one growing season, biennials, which complete their entire life cycle in 2 growing seasons. An example of that would be like dandelion, which just grows a bunch of leaves in this nice little tuft or rosette the first year, and the second year is when it sends up the flowers, and then they die. Perennials grow more than 2 years. They live year after year, and they're woody, and sometimes they're just herbaceous, but they live for longer. More review, plant nomenclature. The first part of a plant's name is the genus. The second part is the specific epithet, or the species. The authority means who named it. L means Linnaeus, a Swedish botanist from the 1800s. He named many, many plants, and that's the scientific name. The species is a population of plants recognized by similar traits in which interbreed and usually have common ancestors too. Let's look at some mixes of different species, like interspecific hybrid, meaning interbreeding between species, different species. I always think of this too, like interstate highways go between 2 different states. Intrastate highways, like the California state highways, go within 1 state. So an interspecific hybrid is between 2 different species. So in this case, this is an example of Prunus pumila and Prunus cerasifera. 2 different Prunus species were crossed, meaning pollen from one was put on the stigma or the female part of another, and one of the results was this hybrid species named Prunus X cistena, purple leaf sand cherry. So that was a hybrid created by humans, and that X means that it's a hybrid, and that's it. And this particular hybrid is sterile. A lot of interspecific crosses are sterile. Animal kingdom example would be like a mule, which comes from a horse and a donkey, 2 different species, interbreed, and you can get a mule. They can breed and they can produce offspring, but the offspring are sterile. So the way to get another mule is you can't just have a mule, breed a mule and a mule. They're both sterile. So you need to get another donkey and another horse to actually breed. Of course, a cultivar is just a word made up joining cultivated variety. It has distinguishing characteristics that make it different from other species, and it has to be cloned and maintained by humans. If you reproduce it sexually, it may not retain those same characteristics because the genetics will change. An example of a cultivar and how you'd write it. Prunus subhirtella autumnalis. That is the Higgin cherry. It's got this beautiful weeping form. So the cultivar name is written in single quotes most of the time, or you could just put CV and then write it with the name capitalized. Legal protection of a cultivar, if you were the one that came up with it or you're thinking about it, you can create a patent, which is good for 20 years. Must be vegetatively propagated, but you can't patent a wild collected plant, but you can grow it in cultivation and demonstrate that you can propagate it, and then you can patent it. You can trademark, and that's just the name that protects the name or the symbol or the logo, and you can collect royalties on plants with trademark names too.